All right, guys. So we left off talking about the myelin sheath, and we have the cells that form myelin sheath in the central nervous system were the oligodendrocytes. Oligo meaning many because the oligodendrocytes could myelinate many or multiple axons simultaneously. And remember, the myelin sheath is basically just a wrapping of the cell. It's not like a separate tissue. It's just that these glial cells actually wrap themselves around the axon multiple times, and it makes many layers that thickens this sort of insulation around the axon, and it prevents currents from leaking out. Now, what we find then is for the peripheral nervous system, each individual segment of myelin is its own cell. These are the Schwann cells, aka neurolemocytes, and they are capable of forming myelin around one cell, or sorry, around one axon, because this is the one cell that's wrapping itself around the axon. That's in the peripheral nervous system. So we know that axons can regenerate because if someone has a damaged nerve, then we understand that there's some recuperation of function. You know, like if somehow maybe you fell with your arm outstretched, it kind of tore some of the nerves in the axillary area, and you might be paralyzed, and then the muscle in your arm might be paralyzed temporarily. We understand that function can be recuperated to some extent, right? So what that suggests then is your axons are being regenerated. But remember, the axon is only a part of the cell, not the whole cell. So if my torso is the cell body, then my arms and legs and stuff would be the extensions of that cell, right? Well, what if I told you all if you cut one, a new one can regrow? Because you're not losing the whole cell, you're just losing a piece of the cell. But the organelles are found in the cell body so that you're able to regenerate these extensions, like axons and dendrites can be regenerated. So as long as the cell body is intact, then the axon can regenerate. And regeneration depends on the extent of damage as well as where the damage occurs. And it's actually the neurolemocytes that help determine how this axon regrows. Could be, yeah, it could be. Like if someone has nerve damage from, an accident of some sort, and they have a nerve that's cut, then you can cut some of these axons. But luckily, some of these axons can regrow, and I'll explain it in the next slide. So what we see here is our neuron cell body, and we have dendrites that come off of it. Now there's an axon that extends off of this neuron cell body, and it goes out to some sort of peripheral tissue, like a smooth muscle, right? So if this neuron's going to smooth muscle, what type of neuron is this then? If it's going to connect to some smooth muscle here. Efferent or afferent? Efferent, because it's going to muscle. And then is it somatic or autonomic? Autonomic motor, because it's going to smooth muscle. Good. So let's just say this axon gets cut. It's cut right in the center, and what that means is these halves don't communicate or connect anymore, and then they can't transmit information to the muscle. Well, that's not the end of the story. Well, what happens then, you guys, the segment of the axon that's cut away from the rest of the cell body, that will eventually degenerate and die away because it's not connected to the rest of its cell, so there's no way for it to regenerate because there's no organelles associated with it, nor are there nutrients being transmitted down the cell because it's cut away from the cell. So what we're left with then is the end of the axon that is attached to the neuron cell body. It has a sealed and swollen end that kind of pinches together. That way it prevents the entire cell from dying. And then the sealed and swollen end starts to follow what I like to call like the breadcrumb trail of fragments back towards the muscle. Because what happens is after this axon dies, it actually leaves behind a chemical signature. So you see all these little fragments of the previous axon. It turns out that this axon can be attracted to those previous fragments, and these neurolemocytes formed a tube. Remember that? So let's think about this. The axon was surrounded or wrapped by neurolemocytes or Schwann cells, right? When the axon dies, then the Schwann cells are still there. So what this means is that there's a tube now where the axon's missing because it degenerated. So what's interesting then is this new axon can flow through the tube of neurolemocytes back to the original tissue. But what this needs to, the way this only can occur then is that this segment of the axon has to be close enough to where it used to be. If it's too far away, it won't find that site, and therefore it won't regrow back to its muscle. So it has to be close enough to the original site of the damage, and there can't be scar tissue in the way either, because if there's scar tissue forms here, it can prevent this axon from actually reaching its target tissue. So what we see here then is after the axon degenerates on this side, the sealed and swollen end of the axon then regrows in the direction of the neurolemocytes, and then finally can attach to its target tissue. And then what we find is the neurolemocytes or Schwann cells start to remyelinate that particular axon. And that's how you can regain some function. And this has happened to me before, you guys, because I broke my nose some years back. And after my nose was broken, they pushed it back into place, like by hand, because the whole thing was smashed off to the side. Like at the time it happened, my friend, I didn't take anatomy yet. My friend was in anatomy, and he yelled, "Dude, your nose is lateral to your face." And I don't know what that meant. I hadn't taken anatomy yet, but it didn't sound very good, right? Because my whole nose was like smushed off to the side. So I went to the ER, and the ER doc basically had to push it back into place. But I think it severed some axons because I lost feeling at the tip of my nose. And it was gone for a while there. I mean, it took months to come back. Now I can finally feel the tip of my nose. I don't even know if this is like the same sensitivity that I used to have. I just know I can at least feel my nose tip, which is good. And what happened then is that after my nose broke, I probably severed some axons too. And then those axons can actually just follow their old pathway along these neurolemocytes back to the skin or muscle from which they attached. It depends on how long the axons have to regrow. And not all these axons will even get to their original place. So this is one of the reasons why, like, if someone has spinal cord injury or nerve injury, they don't always recuperate function, because it's a very delicate process. Like, you're talking about how this has to be matched up just right by the place where it used to be. Like, the axon has to be not too far away, because if it is, it doesn't just know where to regrow, and it will only follow the previous side of axon. So it's got to be close enough. And you can't have scar tissue that forms and blocks away. If scar tissue forms here, and you get a big old thick sheet of fibers, then this axon might not even be able to get through that scar tissue, which means it'll never get to its target tissue. In fact, what happens when scar tissue forms, if it forms, it causes the axon to bump into it, and the axon starts to grow backwards. When axons grow backwards, it actually kills neurons. Like, when axons start growing back, the neurons actually undergo apoptosis. They undergo a process of programmed cell death. And if that happens, then now you're losing the neuron as well. So you have less likelihood of these things even growing back and regaining function. Yes?
So if you have like nerve pain or like tingling or pins and needles, what's like when you have nerve damage, what is that? What's causing that? How is that happening?
I know what you mean, yeah. So sometimes what can happen is when the nerve endings regrow, you can get like tingling sensation or paresthesias, right? Or it might feel like pins and needles. When they regrow, I don't know if there's a simple answer to that. But if I had to guess, it probably has to do with the fact that there's brain areas that correspond with specific nerve endings, and those have to match up again. And so I think it's that sort of abnormal sensation is because there's rewiring that's occurring. And so you're interpreting that as sort of pins and needles, but it's just the rewiring of those neurons. Yeah. So nerves are not the same as nerve cells, you guys. So when you think of like a nerve, like the brachial nerve or the radial nerve or the ulnar nerve, like remember we talked about the ulnar sulcus right here by your olecranon process. If you bump your elbow, that's why it's called the funny bone. Well, your ulnar nerve is very superficial there. The nerve is like the thickness of a pencil. So it's actually a pretty thick nerve. But that nerve is a big old bundle of axons. That's what nerves are. It's basically a big old fiber optics type of cable, and it's just a huge bundle of axons there. So nerves are bundled very similarly to muscle. Remember with muscle we had like epimysium, perimysium, endomysium. Okay. Now instead of mysium we have neurium. So now we have epineurium, perineurium, and endoneurium. And it's basically the tissues that wrap nerve fibers or axons. So epineurium is what surrounds the entire nerve. Perineurium is what surrounds individual nerve fascicles. And then endoneurium surrounds each individual axon.
So,
yeah.
For the endoneurium, it says it wraps
Yeah.
So it wraps even around the myelin sheath.
You got it. Yeah. Yeah, that's a great point. Yeah, endoneurium is not the same as myelin. It's actually around the myelin. And think of endoneurium as more of like a loose areolar type of connective tissue. Yeah, great point. So let's check this out, you guys. And so around the entire nerve, what we find that is epineurium. And then nerve fibers or axons are bundled into little collections called fascicles, just like muscle. Each nerve fascicle is surrounded by perineurium, just like muscle fascicle had perimysium. And then each individual nerve fiber or axon is surrounded by an endoneurium, which is kind of more of a nourishing type of tissue. What's also really cool about nerves is they have blood vessels on the inside. Well, muscles do too. But what's neat here then is that the nerves actually get their blood supply from a vessel you find within the nerve itself. We call this a neurovascular bundle. So what you'll find is often that nerves and blood vessels are bundled together in the same kind of local area. And we start learning about the anatomy of nerves and blood vessels. What we'll notice then is that a lot of times these have the same name. So if there's an ulnar nerve, it'll be nearby an ulnar artery. Okay, if there's a radial nerve, it'll be right by the radial artery. So it actually makes sense that these things kind of are bundled like that. In fact, there are neurovascular surgeons now, where basically a surgeon could go through a blood vessel to help repair a nerve. Because let's say if you wanted to repair a nerve from the inside out, or just get really close to a site of nerve injury, well, you can get there by going through a blood vessel. And if you follow the right pattern of blood vessels, you can get to a specific place where there's a damaged nerve, and then repair that damaged nerve, then back out of the vessel, and then get out of the tissue, without ever having to make an incision in the area where that nerve was cut, which is actually really cool. Talk about the future there. It's pretty amazing. Oh, Danielle, do you have a question?
What in that picture, kind of dividing it into three?
Yeah.
What is that structure?
Well, this is the whole nerve itself.
But I mean, like on the individual.
Oh, this?
One, yeah.
Oh, good question. You know, I imagine that this is still part of the fascicle. Like this is one fascicle and this is another fascicle.
Oh.
They're just kind of showing it as one large bundle here.
Okay.
But I think each one of these is a nerve fascicle. So, you know, this is still some perineurium right here, as well as this would be perineurium going through it. So this would be a separate nerve fascicle. Yeah, great question.
Although it doesn't really show that with the other one. So the other one says where it's labeled fascicle.
Yeah.
That's the whole thing.
From side to side. Yeah. You know, I'd say don't worry about it.
Don't stress.
Yeah. That's a great question, though. Now, we can see our nerve fascicle here, right? So it surrounds each individual. I'm sorry, this is actually an axon. I'm sorry, I take that back. So what's really neat here, you guys, is that this is actually our axon, which has endoneurium around it. We got some blood vessels nearby as well. And these nerve endings, the nerves in cross-section almost look like smooth muscle. And if you look closely, you can actually see the myelin sheath. Like from here to here is one little myelin sheath. From here to here is another myelin sheath. So you can see them up close. Now, where nerve cells communicate, we call it a synapse. And so a synapse is basically where one nerve cell communicates with either another nerve cell or another glandular or muscular cell in the body. And that forms a synapse, which is basically a specialized junction where one cell can communicate with another. And so we have a presynaptic neuron, a postsynaptic neuron, and a synaptic cleft. This is kind of like we talked about last week. The presynaptic neuron is the one that's releasing neurotransmitter. Okay? So an example of this would be like if a motor neuron releases neurotransmitter onto some muscle. And the postsynaptic neuron is the one that's actually receiving signals from the presynaptic neuron. Okay? And there's a space between the two, which is the synaptic cleft. That's a space for a neurotransmitter to diffuse into. Okay? Now, there's different types of synapses. We actually have chemical synapses and electrical synapses. The chemical synapse is the one we've talked about prior, like in muscle tissue, where we have our axon, and that kind of will end up expanding into a bulb here called the synaptic knob or terminal bouton, right? Within that, you're going to have a lot of neurotransmitter-containing vesicles. So last week we learned about how these vesicles could contain acetylcholine, and when these are exocytosed into the synaptic cleft or space, that can bind to receptors on the motor end plate to help initiate an electrical impulse or action potential in the muscle cell, right? Now, let's just say for now This is one neuron synapsing with another neuron. So this would be our presynaptic neuron releasing neurotransmitter onto a postsynaptic neuron that has the receptors, and there's a space between the two called the synaptic cleft. That synaptic cleft is very narrow, by the way. You wouldn't be able to see this under a light microscope. It's very too small. You're talking about how it's in the order of nanometers, and at that scale, you're talking about the wavelengths of light. Like light kind of moves like a wave. Nanometers is in the wavelengths of light. So that means that this space, although this is a space, you guys, this space could be so small that maybe light can't even fully get through the whole thing, which is why you wouldn't be able to see it under a light microscope, which makes sense. So that's a chemical synapse because chemicals are released. What triggers the release of these chemicals from a chemical synapse? Like, yeah, it's calcium. Yeah, good. So when we had a nerve impulse or action potential travel down the axon, it opened up these voltage-gated calcium channels. Calcium rushed in, and then calcium triggered the exocytosis of neurotransmitter-containing vesicles because calcium interacted with those snare proteins, which allowed for exocytosis. And that's an active type of transport, so it takes ATP to do this. Yeah, there's mitochondria there too. Exactly. Very good. Yeah. Presynaptic neuron means it's sort of, the beginning of the synapse, and this is the presynaptic neuron means it's the one that's releasing chemicals onto the next cell. And then the postsynaptic neuron is the one that's receiving the signal from the presynaptic neuron. Now, this is a chemical synapse, but not all synapses are like this, you guys. We also have electrical synapses. Electrical synapses are basically where cells are connected by gap junctions. So going back to like chapter 3 on tissues, we talked about how we had intercellular junctions, and cells could be interconnected by gap junctions. What was a gap junction? Kristen got it. It's a space. It's a tube. Exactly. It's basically a protein space or tube for molecules to diffuse from one cell to the next, right? And so gap junctions were used for communication. So an electrical synapse is when a presynaptic neuron and a postsynaptic neuron can be linked up by protein tubes where electrical current can just directly diffuse straight into the next cell.
Not active transport.
It's not active transport. You got it. It's passive. And these molecules are just spreading right into the next cell. So which one do you guys think would be faster, the electrical synapse or the chemical synapse? Electrical. Why electrical? Why would electrical be faster? Yeah, I mean, okay, so for one, it isn't required to be, there's less steps. Exactly. You got one step, right? That one step is chemicals just stepping in the next cell.
From high concentration to low.
Exactly. Those molecules can just go from high to low concentration or due to electrical gradients. Now, they'll just spread right through. So these are much faster. So if they're faster, what parts of your brain do you guys think would use an electrical synapse? Maybe for pain, like for really rapid transmission of pain. Maybe if you want to speak really quickly, right? Yeah, reflexes. So reflexes would make sense. If they're one of the fastest things that can occur in your body, reflexes would be more likely to have an electrical synapse, right? But why have a difference between electrical versus chemical synapses? Like, why not just have electrical? Why even have chemical synapses? What do you guys think? Yeah. Maybe because this maybe uses more energy? Is that what you're saying? Yeah. Well, this one's actually passive.
Yeah,
so this actually, chemical synapses require more energy. There's an interesting phenomenon, I'll tell you guys, because it's not necessarily an easy answer. Because there are lots of steps in a chemical synapse, it's more regulatable. You can change a chemical synapse better than you can change an electrical synapse. Electrical synapses are way too rigid, and they will only behave in one way, right? Which is to transmit whatever the one cell had directly in the next cell. But with a chemical synapse, you can actually release more or less neurotransmitter depending on the amount of exocytosis you got. So you actually can change the magnitude of the stimulus, right? You might release a little bit of neurotransmitter or a lot of neurotransmitter, right? And what if I told you guys too that these vesicles can also be full of different types of neurotransmitters?
Like so many.
Yeah, so like some vesicles might have one type of neurotransmitter or chemical, another vesicle might have another type, and these vesicles can be released under different circumstances. So like maybe if the neuron's really active, it can release one type of chemical, Or if it's not so active, it will release another.
We have like serotonin.
Yeah.
And,
Yeah.
For the electrical synapse, where's the cleft? Is it the gap junction itself?
There actually isn't. There isn't really a cleft because the cells are almost like they're fused together, right? So these proteins are stuck together. There's not really much of a space. So we don't really talk about there being a synaptic cleft in electrical synapse because the cells are just stuck together. Yeah, Daniel.
So with talking about the chemical synapses and how you can kind of regulate it better, isn't that what some medications like
Absolutely.
SSRIs and things like
Absolutely. Yeah, when you start talking about psychoactive drugs, you guys, they're going to affect these chemical synapses. Whether it's a pharmaceutical or an illicit drug. If it's psychoactive, it's going to affect a chemical synapse more likely than an electrical synapse. So it's really interesting. And, you would learn more about that in like pharmacology. We'll get to a little bit of that throughout our neuro unit here in A&P. But it is fascinating. And when you start changing the properties of a chemical synapse, on a global scale, it can change things like, the amount of sadness someone experiences, right? If they're depressed, you can give them a certain drug that can make them less likely to be depressed, right? Which is actually pretty amazing. So it's pretty